exceedingly good, so I'm really pleased. Although I am sure the university's changed their policy that you now have to get a 40% in each final assessment. It's got something to do with it. But having said that, the marks are good. I'm pleased. You've all done very well. Now, the final assessment is, of course, the final exam. The exam has this structure. I haven't changed the structure of this exam in several years, so it's always the same, and it always has 4 sections. And you are required to answer each section in a new booklet. The reason for that is of course, each section relates to each lecturer, and it's just really easy for us to separate the booklets and give them out and make sure we get them all marked in time for the UTS deadline, which really is quite onerous on our side because the university's deadlines are often quite ridiculous. But anyway, there are 4 sections. There's 1 section for each lecturer. So there is a section from me, 1 section from Alice and Heather, 1 from Tony George, and 1 from Anne Simpson. And you are required to answer 2 questions from each section. Please ensure you answer the 2 questions from each section in the same booklet. Okay? There's nothing worse than actually getting all the questions in 1 booklet, which has happened, so I can assure you it really irritates me. So do get those basics right. There is a simple calculation in the final exam, so we do allow you to use a calculator, but you do not need a calculator for the questions. Because in my experience, it's all very simple calculations and you can work them out in your head. Yes, each section has 4 questions, so you have to answer 2 of the 4 questions. And of course, the rules are now you've got to get a 40% pass in this exam to pass the subject. The 50% in the book, in the manual is actually wrong. So, what did I cover in my lectures? Well, the idea was to convey the concepts of genomics, the technical basis of genome sequencing, the ideas of how a genome is structured, and the types of sequences that you find within a genome, the idea of gene families, repeats, and so on. And I would hope that you would have got the idea that some pretty basic definitions are needed in terms of what is a genome, understanding what is a transcriptome, what is a proteome. And I have asked similar questions in the past. Define these terms. Genomics. So I covered the idea of what is genomics, the idea of shotgun sequencing for sequencing a genome, and I tried to emphasize not only in the lectures but also in the practical sessions the importance of computer-based biology, the importance of bioinformatics, and that ultimately you can do something about understanding functional and comparative genomics. I did try and emphasize with the idea of repetitive sequences. You covered repetitive sequences in the lectures. You've done a crack on repetitive sequences. You should be able to tell me about repetitive sequences, the idea of highly repetitive sequences, moderately repetitive sequences, unique sequences. There are different groups of sequences, mini-satellites, micro-satellites. Describe the different sequence classes that we recognize in a human genome. And I touched upon ethical issues as part of that process of talking about genomics. For me, one of the most important things that you should understand is the ideas behind the Human Genome Project. What are the aims of the Human Genome Project? How was it accomplished? What were the outcomes? were the most important outcomes, such as genome size, numbers of genes, and the importance of alternative splicing as a way of generating larger numbers of proteins from a set number of genes. I would hope that you would be able to explain to me, to anybody else in your family, your friends, what were the benefits arising from the Human Genome Project. Now think carefully about this one, because in the past when I've asked this type of question, I've had answers that have talked about engineering microbes to clean up oil spills and stuff like this. But, you know, the reality is the Human Genome Project, we're talking about human medicine here.
Yes.
So what were the structures of genomes? What is the structure of genomes? We touched upon DNA content, genome size, complexity, the ideas of mutations and duplications, the ideas of sequence classes, that there are pseudogenes, repeats, different types of repeats, transposable sequences.
Just take photos.
We looked at gene structure. We looked at the idea that different parts of a gene structure has different functions. We looked at exons and introns, both in the practical classes and in the lectures. And the idea that you can recognize different parts of a sequence that contributes to the gene structure. So hopefully you have some idea that this is the structure of the eukaryotic gene, that you can recognize exons separated by introns, and that through alternative splicing you remove the introns to generate a messenger RNA that is translated at the ribosome. Okay? Almost an immediate fail in molecular biology too is mixing up the terms transcription and translation. When I see this, I immediately deduct about 50 marks from every question, because, you know, you're third year students now. DNA makes RNA, makes protein. DNA is transcribed to give RNA that is translated at a ribosome. You don't deserve to pass if you can't tell us that. In the practical sessions, you spent a lot of time looking at a set of genes known as the homeobox genes that are important transcription factors and therefore central in regulating mammalian development. Not just mammalian development, development of most eukaryotic organisms. Most eukaryotic organisms have homeobox genes. The idea that you could have constitutive and inducible expression, that these transcription factors facilitate that process by initiating and regulating transcription, and that alternative splicing can give rise to different protein isoforms that may have different functions in different tissues. And I've tried to emphasize the importance of these genes by asking you to look for associations of changes in expression of these genes with disease, and most of you managed to come up with at least one or two diseases that were associated with homeobox genes. But at all times through the practical sessions, I tried to get you to write a paragraph about the biology of these genes, because they are important genes. They do convey central meaning and functions about how gene expression is regulated. Now, Dr. Heather was asked to continue the same similar dogma of looking at transcription and regulation of transcription in eukaryotic cells. So here we still have the central dogma that DNA is transcribed to give RNA that is translated to give a protein. And the important thing to note in the eukaryotic organisms is that part of that regulation is facilitated by having different components of that process in different compartments of processes in between that actually regulate expression as well as turning on and off transcription. So we understand the genes are located in the genome, the genes make up a small proportion of the DNA. Essentially, the exons are the important bits, introns, and that all that process is regulated by a promoter. The promoter, of course, is the place where essentially RNA polymerase binds, and therefore it is critical for turning on and turning off transcription. Now, in order for a gene to be turned on or off, clearly you have to do something about the wonderful supercoiling of DNA around the promoter and other parts of the gene. And, of course, that supercoiling results because the DNA is tightly wound, it's complex with a wide range of proteins, like histones, to give you the basic unit that we know as a nucleosome. And Dr. Heather went through some core parts of the different regulatory process. She emphasised the different components of what is a promoter, the idea of a tartar box was covered, and also that expression may be due or regulated as part of a set of response elements. And she talked about the idea that different transcription factors behave in slightly different ways. They have different sequence recognition elements, response elements that you need that are used for control of gene expression. So there's the example there, the SPR1 binding site has a core sequence, a consensus sequence that is essential for this type of gene expression. Another set of well-known regulatory sequences are what are called enhancers that alter transcription. They may or may not be located close to the genes of interest. They may be quite a long way away from the genes that they are influencing. But again, they contain some kind of response element, a regulatory element that influences gene expression. Dr. Heather also talked about how are transcription factors regulated. But there's all sorts of other mechanisms that are involved there, phosphorylation of transcription factors, DNA methylation, ligand binding, and so on. DNA methylation is quite important in eukaryotes, as you can see from the recognition that you have large CG islands scattered around the human genome, the eukaryotic genome, and essentially, methylation is normally done at certain bases within the genome, and in this case, we usually talk about the CPG dinucleotide that is methylated on the cytosine residue. So the idea of CG islands being that there is lots of methylation at those CG sites. So we now recognise that CPG islands occur extensively around areas that need to be regulated, and the idea that methylation can influence gene expression is now universally accepted. RNA processing. Clearly, if you have a gene that is being transcribed to give RNA